So this is part 2 of chapter 12's lecture on the cell cycle and mitosis. So where we left off was on interphase, and just finishing that up, just to remember some of the characteristics of interphase is during this phase the nucleus is well defined and the DNA is loosely packed in long chromatin fibers. This is the phase when preparation for mitosis is occurring. It's when the S phase, when replication of the chromosomes occurs, and it's when also the work of the cell is going on, the production of proteins and organelles. And again, in preparation for mitosis, that production of organelles is occurring specifically for mitosis. Interphase is also when we can have a cell go into its resting phase, which would be that G0 phase. So now a portion of interphase that we're going to focus on right now is called the S phase, or the synthesis phase. This is when the copying or replicating of DNA occurs. So this is when within the nucleus, we have to separate the DNA strands to make a copy so that we get 2 copies of the DNA. And human cells duplicate about 3 meters of DNA, and each daughter cell gets that complete identical copy. They have figured out that the error rate is about 1 error or mutation for every 100 million bases. So the typical mammalian genome has around 3 billion base pairs. So that means every time we have a cell cycle, every time we have a mitosis occurring, we get about 30 errors. So those are mutations that are going to provide some diversity in terms of the genetic or gene pool that can occur with those daughter cells. So now organizing DNA, this is again just a little bit of a repetition, but focus again that DNA is organized into chromosomes. We have a double helix of DNA molecule, and that DNA molecule is wrapped around histone proteins. And so that's how it's actually stored when it's not being replicated or when it's not being transcribed. So you can think of histones as kind of spools, and then the DNA is thread, so that wrapping around the histone is like seeing thread around a spool. And there are a number of histones for every chromosome. And a definition of that DNA and protein complex, the histone and the DNA together, is defined as chromatin. So during mitosis, the chromatin is actually what we see as it becomes condensed. That little bottom picture there shows a duplicated mitotic chromosome. And that's not to be confused with a homologous chromosome. So remember that a homologous chromosome, going back here, a homologous chromosome means that we have, if we have, for example, in humans, 2 sets of chromosomes, one set it comes from the mother and one set comes from the father, and together that set, they're not identical, but they do contain the same types of genes on each chromosome. So that set is what makes up the homologous pair of chromosomes. And that is different from what we get at the end of the S phase of mitosis. At the S phase of mitosis, we have 2 identical chromosomes. So we have 2 sets of DNA that are exactly the same. And this is a picture showing the results of the replicated DNA. So So this is the two sister chromatids that are the identical copies of the same chromosome. This is another picture showing that process. So we start out with one chromosome, one of the homologous pairs, and then through duplication, through replication, we end up with the two sister chromatids. Those two sister chromatids are joined together in the middle by what we call the centromere. And we kind of think of the centromere as like a narrow waist of the duplicated chromosome, where the two chromatids are most closely attached. So along the two sister chromatids, they're actually both, they're bonded together all along the edges of the two sister chromatids. But the most closely held together region is at that centromere. And the centromere is also where the kinetochore forms, which is where the attachment to the mitotic spindle is going to occur. So that's the importance of that centromere, is one, to hold those two sister chromatids closely together, and two, to give a place for the kinetochore to form, which will attach to the mitotic spindle. Here again, we've seen that same idea. And what this is showing there on the bottom with that purple and the green on the lower right is showing the two homologous chromosomes that you would find in a person's nucleus inside their DNA. So the purple, let's say, is from the mother, and the green is from the father. So they're two different chromosomes coding for the same proteins and coding for the same genes. Homologous means the same information. Each of those, if we are going through replication, then we get identical copies of each one. So as you can see here in the picture, going from homologous chromosome to sister chromatids that are together, they're from left to right. So, or single-stranded versus double-stranded. So this is an overview of the mitotic process, and we'll show this really quick here.
This sucker flyer's meat will be as good as new in a few weeks. Thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
So this short little video is from your textbook, from the Pearson Publishing. And it's a good overview as we move into looking more carefully at each of the phases of mitosis. So we're going to look at each phase, starting with prophase. And each phase has some key characteristics that you should keep in mind. For prophase, there are two. One is that the nucleolus disappears within the nucleus, and also that the nuclear membrane breaks down. And as you saw in the video, there's still that condensing occurring of the different sister chromatids. Another thing that's happening during prophase is that those centrioles are moving to the opposite poles of the cell. And remember that centrioles are only found in animal cells. Also, we have the mitotic spindle forming, and that's part of that process of those centrioles moving to the opposite poles. That mitotic spindle is made up of microtubules, and those microtubules are made up of actin and myosin, just like our muscle cells. The mitotic spindle, again, coordinates movement of the chromosomes, and you guys should know that. The next phase is called prometaphase, and this is that transition phase between prophase and metaphase. Its key characteristic is that the chromosomes begin to move. As that video again was showing, that movement is helping them get to that central portion, that what we consider the metaphysic plate. And what's occurring now is those spindle fibers are starting to attach to the centromeres, creating that kinetochore, that little protein connection that is occurring between the centromere and the mitotic spindle. The next phase is metaphase. We think of that as, I always think of metaphase as the meeting up phase. That's when we have the chromosomes lined up across the middle of the cell. So the key characteristic to remember of this phase is that the chromosomes are lined up. And we call that the metaphase plate. Meta means middle. And the spindle fibers, again, are very active in coordinating this movement. And it's important for this phase to occur because this is going to enable those sister chromatids to separate properly so that each new nucleus is going to receive only one copy and one complete copy of each of the of each particular chromosome. So we'll stop this right here, this phase 2 of our chapter 12 lecture, and continue on with part 3